Hello, are there any questions? Can somebody please confirm that you can hear me?
We can hear you.
Thank you. All right, if you didn't notice, I did email out the your unknown number. I did get it mailed out very late Tuesday night. It was actually a little after midnight, but only a few minutes after midnight. What is that? I have to look at that. I imagine you can't see my screen yet. Can close that. I guess I'll leave that there. Where is my... Here it is. I blow this up. Show you the screen now. My computer's kind of slow. Not sure why. I don't have that much open. Oh, I can shut that down. I'll leave that open for the lab. I got a few more things graded. I don't remember exactly what, but I did get all of lab 00 and lab 01 graded. Any questions about the grading? All right, if not, let's begin. I'm going to leave that open because I got to look at that. Let me remind you, since I've got that there, that you do need to put in what you're going to do your infectious disease project on. You cannot simply choose a project. You have to clear it with me. It is going to be due Saturday, August 13th, the infectious disease project. All right, today is that July 28th? And we are going to be, gosh, we need to finish chapter 4 and begin chapter 5. We're a little bit behind schedule. I didn't realize that. Or did we start chapter 5 last time? Anyone remember? Is that metabolism? Yeah, we did start this. Never mind. Excuse me here. Hopefully you didn't see any of that. Let's go to where we were. So we had finished talking about enzymes last time. And you note that enzymes are biological catalysts. They speed up biochemical reactions. They are generally specific for one molecule, the specificity being the three-dimensional function or shape of the enzyme. And then it binds specifically to, and we're going to oversimplify it, but in this class we're going to say it binds specifically to one substrate. And then the enzyme actually does form a complex with its substrate. Any questions about any of that? All right, enzymes normally end in ACE or ASE. So most enzymes, if you see it, like transferase, hydrolase, catalase, we'll run into catalase in the lab. So most enzymes end in ACE. You don't need to know any of the examples. There are some enzymes that do not end in ACE. They're generally the older enzymes like renin, but most enzymes end in ACE. This is a convention for naming enzymes. You should also note that some enzymes don't work alone. You don't need to know the name, but we have an enzyme and it gets together with either a coenzyme or a cofactor. A coenzyme is a small biological molecule like NAD+. We'll talk about that in this lesson. It's a coenzyme and it gets together and kind of finishes the enzyme. So now the enzyme can bind to its substrate. A coenzyme differs from a cofactor, and that is a cofactor is usually small, a non-protein portion, helping out the enzyme. And many coenzymes are just elements like iron, zinc, magnesium, calcium. So a coenzyme is a biological molecule. A cofactor is just an element, meaning an atom of one element. Then it gets together to finish the enzyme. You don't need to know the names of the different parts of the enzyme. We will talk about a couple of coenzymes in this lesson. NAD+, also called nicotinamide adenine dinucleotide. You don't need to know this, but it's a niacin derivative. It's primarily involved in catabolic reactions. We will talk about aerobic respiration, in which case NAD+ will be mentioned. We'll also talk about FAD, flavine adenine dinucleotide. It's a riboflavin derivative, and it will also talk about anaerobic respiration. Some other important coenzymes, which I guess you don't really need to know, just that there are different important coenzymes. But NADP+, similar to NAD, green plants have this coenzyme. FMN or flavine mononucleotide. And then coenzyme A, which I'm not going to require you to know, but that is a coenzyme. But actually, we will briefly talk about coenzyme A in this lesson, later in this lesson. So you do need to know the first two. We'll talk about them in detail with aerobic respiration. And those are the only coenzymes you need to know. So let's move back to talking about enzymes and how they work. An enzyme can be said to work like a key and a lock, or called the lock and key model, where an enzyme specifically fits to one substrate and then converts that substrate into the products, much as a key fits into a lock and then can unlock the lock. And the idea is that the lock and key model, there's only one key that can fit the lock, and there's only one enzyme that can fit the substrate and then convert it to its product. That's the lock and the key model. The other model is the hand in the glove model, which I'll talk about in a little bit, but I want to show you it's a little bit more of a true aspect of an enzyme. I'll go ahead and tell you what the lock and the hand in the glove model is. And that is an enzyme binds to a substrate much as a glove will bind or fit over a hand. And that is the glove will mold to the shape of the hand, so it will then fit the hand. And that's much as an enzyme will mold to fit the shape of a substrate. What do you notice about this picture here? Here we have the substrate and the enzyme. And then we have the enzyme substrate complex where they're bound together. What do you notice about this? There's actually three things you can notice about this. Come on, don't be shy. I've already told you one of them. Nobody's going to take a guess? Well, I'll tell you the one I already told you, and that is the enzyme changes its shape to bind to the substrate. And I'll talk to you about the third one. I'm not going to tell you the second one. You guys are going to have to guess it. But if you look very closely at this substrate here and here, you'll note the substrate is changing its shape when it binds to the enzyme. And that is the substrate is a little flatter here than here. It's a very slight change, but it is demonstrating, see if I can blow that, shrink it down now. It is demonstrating the stress on the chemical bonds in the substrate that occur to the substrate when it's bound to the enzyme. And when you put stress on those bonds, it encourages the substrate to change into the products or product. And so even the hand in the glove model isn't quite correct because it's more than just the glove molding to fit the hand. In this case, it is the glove or the enzyme is molding to fit the substrate, but the hand is molding to fit the glove. And you can think of it as a shapeshifter putting his hand in the glove, where the glove is molding to fit the shape of the hand. And then the shapeshifter's hand, oops, didn't mean to do that. The shapeshifter's hand is molding to fit the glove. And those of you who have watched Star Trek Deep Space Nine, you know what I'm talking about. Any question about any of that? All right. Let's move on. Enzymes do not work in a vacuum. You already know that some enzymes require either a coenzyme or a cofactor. Well, enzymes require other things to work. They don't work in a vacuum. They do require the correct temperature, the correct pH, the correct salinity, generally the absence of heavy metals, the absence of alcohol, the absence of UV radiation. And when any of these are present in the correct or incorrect concentration, that can cause protein denaturation. Protein denaturation is where, for example, the temperature is not correct, and so the bonds holding the enzyme together, largely hydrogen bonds, but it could be ionic bonds, can break, and then the enzyme unfolds and takes on a different shape, and that we call protein denaturation. And if you put the enzyme at the wrong temperature, you can not only break certain hydrogen bonds, like when you heat up an enzyme, you break the hydrogen bonds, but when you freeze it, you can have hydrogen bonds forming that should not form, and that can also denature the enzyme. And that can be true when the enzyme is in the incorrect pH or the incorrect salinity. The enzyme can either break bonds that should be there, or it can form bonds that should not be there, and then the enzyme will unravel and come out of the correct shape. Heavy metals tend to denature proteins or enzymes. Alcohol denatures proteins or enzymes. And then UV radiation does the same thing. So enzymes don't work in a vacuum. Enzymes also do require substrates to work. Like if you had an enzyme in a vacuum, there's no substrate for the enzyme to work on. And so the enzyme isn't going to speed up the chemical reaction. You do need substrate concentration, and the optimal enzyme reactivity, meaning the enzyme will react at the optimal or the best rate when you have enough substrate present. We'll talk a little bit more about that. Enzymes don't usually work when there are enzyme poisons present, like cyanide and fluoride. They can stop the enzyme from working. Usually, like with cyanide, the poison will denature the enzyme, and then it'll unravel and take on a different shape, stopping the enzyme from working. Any question about any of that? There's also inhibition of enzymes, where we can have something which we'll call an inhibitor, which can stop the enzyme from working. There are two types of inhibition. There's competitive inhibition and non-competitive inhibition, which is also called allosteric inhibition. And then lastly, we'll talk about feedback inhibition. Any question about any of that? All right. So I mentioned enzymes have to have their proper shape for the enzyme to be functioning. And this is a protein enzyme, but even with an RNA enzyme, it does have to have a proper shape and the proper folding and binding patterns. And then when the enzyme is denatured, it'll be in a different folding pattern. Enzymes have to be in the correct temperature. This enzyme is an enzyme that might work in a person because it works optimally at 37 degrees. And that would be most of your enzymes. Maybe the enzymes on the skin would have a slightly lower temperature than 37, although it wouldn't be that much lower. A few degrees lower would be the enzyme working on your skin. And so this could be, like I said, a human enzyme, and it works best at 37 degrees Celsius. That's 98.6 degrees Fahrenheit. And when you move away from that temperature, the enzyme will work less well, the enzyme activity increasing on the y-axis, temperature on the x-axis. And as you go further and further away, the enzyme will have less and less activity until you've totally denatured the enzyme. In this case, when you've heated it above 50 degrees Celsius or lowered it less than 10 degrees Celsius. So that would be putting the enzyme in the refrigerator. And then human enzymes aren't going to work in the refrigerator, as well as human enzymes would not work above 50 degrees Celsius. I don't know what that is in Fahrenheit, but it's getting very warm. The same thing with pH. This enzyme works best around pH 5. Does anybody know where we would have an enzyme that would work best around pH 5? Come on, somebody must have a guess. Most of our enzymes work best around pH 7 because That's the temperature or the pH of most of the cells in our body. Like our blood, it's a little over 7. I don't remember if it's 7.4 or 7.3, but the pH of the blood is slightly higher than pH 7, like 7.4, but still the enzyme's close to 7. When would we have an enzyme around pH 5?
Would it be within our digestive system?
That would be a good guess, but no, our stomach will be closer to pH 2. And so this enzyme isn't working best at the pH of our stomach. Excuse me. That's probably from the air conditioning system. So like renin, an enzyme that is active in our stomach, will work best around pH 2. This would probably be an enzyme in our skin. Our skin is pH 5. And we'll talk a little bit about why the skin is pH 5, but it's mainly to discourage a human pathogen from growing on your skin. Because it's pH 5 and most human pathogens want to grow at pH 7. The point being that enzyme have an optimal pH, and when you go away from that pH, the enzyme will have less activity. And then when you go way away from that pH, the enzyme will have no activity. At pH 10, this enzyme is denatured and will have no activity. For substrate concentration, when you have no substrate present, the enzyme can't work because the enzyme has to bind to the substrate to perform the chemical reaction. So there will be no chemical reaction if there's no substrate. As we increase the substrate concentration, you'll note that the enzyme activity greatly increases. But then the enzyme activity starts to slow and then slows further and then plateaus off. Where when it reaches the plateau, when we increase the substrate concentration further, there is no increase in the enzyme activity. Why would that be? Why, when you increase the substrate over here, it does not increase the enzyme activity? Nobody has a guess? Well, at this point, all of the enzymes are already bound to the substrate. And so if you increase the substrate concentration further, you get no further increase in enzyme activity because all of the enzymes are bound. They can't work any faster than they're currently working. Okay, any question about that? Whereas here, when you increase the increase the substrate concentration, that means the enzyme is more likely to bind to the substrate, and we increase the enzyme activity. Why the enzyme activity increases very quickly here when the substrate concentration is very low. This is also true when you have increased the amount of enzyme. We don't have that shown here, but if you have the substrate concentration at a certain concentration and the enzyme being 0, there will be no enzyme activity. And then when you increase the enzyme, the enzymatic activity will increase up until a point where all of the enzyme is already bound to the substrate, in which case increasing the enzyme concentration further will not increase the enzyme activity. All right, any question about any of that? If not, let's move on to the factors influencing enzyme activity. You can have a competitive inhibitor influencing the amount of enzyme activity. The inhibitor competes with the substrate for the active site if the inhibitor is a competitive inhibitor, meaning the substrate binds to the active site of the enzyme, and then the enzyme can convert the substrate to the products. But if you have a competitive inhibitor, it binds to the active site, and then the substrate cannot bind to the enzyme. And therefore, no enzyme activity is seen, and no substrate is converted to the product. If you have enough competitive inhibitor, because it's competing with the substrate for the active site. That's how a competitive inhibitor works. It binds to the active site of an enzyme, and then the substrate cannot bind to the enzyme. There are also non-competitive inhibitors, also called allosteric inhibitors. And these inhibitors bind to the enzyme, but that binding site differs from the active site of the enzyme. It's called an allosteric site. What happens is the non-competitive inhibitor, when it binds to the enzyme, it changes the folding pattern of the enzyme, which then changes the shape of the active site. And now this enzyme cannot bind to the substrate because the active site is not in the correct shape. And that's how a non-competitive inhibitor works. Also called an allosteric inhibitor because it binds at an allosteric site. Any question about any of that? If not, let's talk about feedback inhibition. Feedback inhibition is a type of non-competitive inhibition or allosteric inhibition. With feedback inhibition, we have an enzymatic pathway. I need to shrink that down just a little bit, right there. Maybe I can blow it up this way. Yeah, that's better. Where the substrate is binding to the enzyme 1, and then enzyme 1 is converting the substrate into intermediate A. We then have enzyme 2 that binds to intermediate A and converts it to intermediate B. And we have enzyme 3, which converts to intermediate B and changes intermediate B into the end product being the last product of the enzymatic reaction. With feedback inhibition, we generally have the end product binding to the first enzyme and then acting as an allosteric inhibitor, binding to the enzyme, changing the folding pattern of this enzyme so that the active site can no longer bind to the substrate. That inactivates enzyme 1, and then no intermediate A is being made because enzyme 1 is inactivated. And then no intermediate B is going to be made. So no end product is going to be made. Any questions about any of that? Okay, that talks about the shutting down of the enzymatic pathway. How do we begin the enzymatic pathway? Well, in a cell that has made all the end product it needs, the end product will bind to the first enzyme and inactivate it. That part we know. But later on in the cell, it might start using the end product, or the end product simply becomes degraded with time. And so the cell will not get enough, will not have enough of that end product. If there's not enough of the end product around, then if there's any free enzyme 1 around, there won't be enough end product around to bind to it and inactivate enzyme 1. So enzyme 1 will be active. It'll take the substrate and make intermediate A, and then intermediate B will be made, and then the end product will be made again. So that way the cell can make more end product. Any question about any of that? All right. No questions? Well, let me add that how does the end product come off of, come on mouse, there we go. How does the end product come off of the enzyme once it is bound, so that enzyme 1 can become active, meaning if there's not enough end product around, we want to make more end product. And so this end product needs to come off of this enzyme. Well, what happens is the binding of the end product to the enzyme is not a permanent reaction. It's actually a stoichiomic reaction. So every once in a while, the end product comes off of the enzyme. And that makes free enzyme 1. If there's lots of enzyme around, another end product will bind to the free enzyme and inactivate enzyme 1. But if there is not much end product around, then it will not bind to enzyme 1, and then enzyme 1 will be active to make intermediate A, make intermediate B, and then make more end product. And that explains how feedback inhibition works. The reason why in our cells feedback inhibition is usually occurring with the end product binding to the first enzyme and inactivating the first enzyme is because this is the energy most efficient means for engaging in feedback inhibition. If you were to inactivate enzyme 2, for example, it would work, but you would make intermediate A, and that takes energy for the cell to make intermediate A, as well as a little bit of time. And so that is not as energy efficient as shutting down enzyme 1. Everyone see that? You see why feedback inhibition usually has the end product shutting down enzyme 1, because it's energy-wise the most energy efficient. In our cells and the cells of microorganisms, or any cell for that matter, usually engages in feedback inhibition with the end product shutting down enzyme 1. All right, if there are no questions, let's move on. I need to talk about ribozymes. I also already mentioned that when we're talking about enzymes, we can have a protein enzyme, which are most of the enzyme. Most enzymes are proteins, but there are some enzymes which are not proteins, and they are RNA molecules, where RNA becomes the enzyme. These have a specific name, ribozymes, and it just means RNA enzyme. And this can be an RNA that cuts and splices RNA, or it could be an RNA that cuts another molecule. It does function as an enzyme in that the RNA enzyme has an active site that actually binds to its substrate. And then after the RNA enzyme performs the chemical reaction, the RNA enzyme is not used up or modified so that it can go on and bind to another substrate and perform countless enzymatic reactions. Any question about ribozymes? There's only one ribozyme that we're going to talk about in this class, and I think we'll talk about it later in this lesson, and that is a ribosome. When it is taking the amino acid off of tRNA and binding it to the growing polypeptide chain to make the protein that the ribosome is doing. The enzyme that's performing that enzymatic reaction is actually an RNA enzyme. So it's an RNA portion of the ribosome, which is taking the amino acid off of tRNA and then binding it to the growing amino acid chain that'll later become the protein when the ribosome is making proteins. That's the only RNA enzyme we'll talk about in this class. Any question about any of this? All right. Let's move on to metabolic diversity. All organisms, including microbes, have metabolic diversity among them. All microbes require an energy source and a carbon source, and they have diverse energy sources as well as diverse carbon sources. We can classify an organism upon its energy source. Organisms can be classified as a phototroph if they derive their energy source from light. So a phototroph gets its energy from light, and that's usually sunlight, but it doesn't have to be. And that's different from a chemotroph, where the organism derives its energy source from a chemical compound. We are a chemotroph. We get our energy needs from an organic chemical compound, such as glucose. But there are some microorganisms which can get their energy needs from an inorganic chemical compound. And we'll talk a little bit about them later. They're the chemosynthetic bacteria, or the chemosynthetic archaebacteria. And they actually get their energy needs. You don't need to know this at this point, but they get their energy needs by stripping an electron from an inorganic chemical compound, such as iron, or a sulfate, or a nitrate. By the way, when we get our energy needs from glucose, we too are stripping electrons from glucose to get our energy needs. And we'll talk a little bit more about that when we're talking about aerobic respiration. Any question about any of that? All right, organisms can be classified on their carbon source. If the organism can get carbon from CO2, either in the air or the water, we call them an autotroph. These organisms can make their own food from CO2 in the air or the water. And of course, they need water in addition to CO2. We're not going to talk about water, at least not yet. But those are autotrophs. They get their carbon from CO2. A heterotroph gets its carbon from an organic chemical molecule. And we are a heterotroph. Once again, we tend to get our carbon from glucose, an organic chemical compound that gives us our carbon. Any question about any of that? Now, if you ever get confused when we're classifying organisms, just break it down into the simplest way, and that is, if it gets its energy need from light, it's a phototroph. If it gets its energy needs from a chemical compound, it's a chemotroph. And if it gets its carbon from CO2, it's an autotroph. And if it gets its carbon from an organic chemical compound, it's a heterotroph. However, these four trophs can be combined, and then we classify organism on, I guess, two characteristics: where they're getting their energy needs and where they're getting their chemical compound. And we have photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. When we classify organisms on where they're getting their energy need and at the same time where they're getting their carbon needs. Let's talk about the trophs in this slide here. The photoautotroph is one that I think all of you are familiar with. So this is a good place to start. A photoautotroph gets its energy need from light, why it's called photo, energy needs from light. And then it gets its carbon source from CO2, why it is an autotroph. And this is familiar to you because green plants are photoautotrophs. They get their energy needs from light and their carbon dioxide, their carbon from carbon dioxide. There's more photoautotrophs, though, than just green plants. For example, the algae, which are very similar to plants, are photoautotrophs, as well as the photosynthetic bacteria, such as the green bacteria or the cyanobacteria, which is actually called blue-green algae, and it's a misnomer because it's not really even algae, but it is blue-green in color, and that would be the cyanobacteria. There are other bacteria which are photosynthetic, and we're not going to talk about them at any length, but most photoautotrophs produce oxygen, like green plants produce oxygen, but they don't have to produce oxygen. There are some photoautotrophs which are anoxygenic, meaning they do not produce oxygen, such as the green and purple sulfur bacteria. They are photoautotrophs, but they do not produce oxygen. And I'm sure none of you are familiar with the green and purple sulfur bacteria. I've actually never even worked with any of these. Any question about any of that? All right, I'm going to skip these two now and move down to the chemoheterotrophs. This should be familiar to you because we are a chemoheterotroph. We get our energy needs from a chemical, and we get our carbon source from a chemical compound. And actually, we get our energy needs and our carbon needs from typically the same organic chemical compound, such as glucose. Oftentimes, a chemoheterotroph will get both their energy source and their carbon source from the same molecule. And all animals are that way, as well as people. Protozoa are chemoheterotrophs, fungi, and many bacteria, not all, but many bacteria are chemoheterotrophs. For example, E. coli is a chemoheterotroph. Now, with that stated, chemoheterotrophs do not have to have the same molecule as both their energy source and the carbon source. For example, is it Halobacterium? Gets its carbon source from glucose, but can get its chemical, its energy need from a chemical compound, in which case it comes from a inorganic chemical compound. This could be sulfate or nitrate or iron. And we'll talk a little bit about that. Actually, I'll talk a little bit about it now. Some of the chemosynthetic bacteria get their energy needs from an inorganic chemical compound, and they're stripping an electron from the inorganic chemical compound. You actually are familiar with one chemosynthetic bacteria. Now, is that a chemoautotroph or a chemoheterotroph? Well, I'm not positive of that, but we'll say that you are familiar with one chemosynthetic bacteria, and that is when you take the lid off of the toilet tank, not the toilet bowl in the bottom, but the water above in the tank. You know, all that brown sludge on the back of that toilet tank? That's actually a chemosynthetic bacteria. Any question about any of that? Oh, and I guess the point is where that bacteria is getting its energy needs are from an inorganic chemical compound that is dissolved in the water. Oftentimes in the water of the United States, it is oftentimes iron, but it doesn't have to be iron. It can be getting its energy need from another inorganic chemical compound, such as if you have well water, you'll be getting, or you'll be having chemosynthetic bacteria growing or getting its energy need and then growing from getting the energy from maybe a sulfur compound dissolved in the water. But it could be another chemical compound dissolved in the water. Any question about any of that? All right, if not, let's talk about the chemoautotrophs. These are organisms which get their energy source from a chemical, inorganic chemical compound, such as hydrogen sulfide, gas, or a nitrite compound, or iron, for example. And what they're doing is they're stripping electrons off of this chemical inorganic compound. They are getting their carbon source from carbon dioxide. And that is why they're called a chemoautotroph. They're an autotroph because they are getting their own carbon dioxide and making their own food from carbon dioxide and water. However, these are not photoautotrophs, they're chemoautotrophs. And some of the chemosynthetic bacteria are chemoautotrophs, and others of the chemosynthetic bacteria are chemoheterotrophs. Any question about any of that? All right, I've got the iron oxidizing bacteria. This would be the bacteria in the back of your toilet tank, which is a chemosynthetic bacteria, which is a chemoautotroph. Any question about any of that? All right, now let's talk about the hardest one, the photoheterotrophs. They are phototrophs, so they get their energy needs from light. However, they do not get their carbon needs from carbon dioxide. They actually get their carbon needs from an organic chemical compound, such as glucose. You probably are not familiar with photoheterotrophs because there are not many of them, and almost all of them are bacteria. An example is the green and purple non-sulfur bacteria. And I'm sure none of you are familiar with the green non-sulfur bacteria. All right, any questions about any of this? Once again, if you get confused on dividing an organism into 4 different trophs, make it a little simpler and go back to, is it a phototroph? Is it a chemotroph? Is it an autotroph or is it a heterotroph? Meaning break it down to its energy source and break it down to its carbon source. And then once you have that, come back to putting all 4 together to answer, is it a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. All right, any questions on any of that? So you should know the classification system shown in red, that all organisms can be classified on their energy source. If they get their energy needs from a chemical compound, they're a chemoheterotroph. If they get their energy needs from a light, they're a photoheterotroph. You then can classify the organism for where it gets its carbon source. If it is a chemotroph and it gets its carbon source from an organic chemical compound, it's a chemoheterotroph. If it is a chemotroph but gets its carbon source from CO2, it is a chemoautotroph. If it is a phototroph and it gets its carbon source from an organic chemical compound, it is a photoheterotroph. And if it gets its carbon source from CO2 and it is a phototroph, then it is a photoautotroph. Any question about any of that? All right, you do not need to know beyond the red line, but let me briefly state that we can further divide the chemoheterotrophs into those which are those that we engage in aerobic respiration, and they use oxygen. And that's happening is where the final electron goes to. The final electron acceptor is oxygen, the electrons go there, and they do require oxygen to engage in respiration, but that doesn't have to be the case. We can have a chemoheterotroph, which goes to a molecule which is not oxygen, meaning the electron flows to a molecule which is not oxygen. And of these, we have two types. The organic chemical compound, the electron goes to an oxygen molecule, which is an organic chemical compound. And the bacteria which engage in fermentation do this. And I suppose your muscle cells, when they don't have enough oxygen, can do this as well. Other bacteria which have their electrons flow to a molecule other than oxygen and the molecule goes to, or the electron flows to an inorganic chemical compound. These are the organisms which engage in anaerobic respiration. And we'll talk a little bit more about that. I guess the other thing to point out is with the phototrophs, you can have oxygenic phototrophs, and they make oxygen, like green plants. Cyanobacteria engage in photosynthesis for their phototroph, and they make oxygen. We do have phototrophs which do not make oxygen, and that would be anorexic photosynthetic bacteria. All right, so let's go back to energy production. Energy is associated with the electrons, which are found in all molecules, but specifically we're talking about the electrons in the chemical bonds of a nutrient molecule. The reason why humans need to eat is we need to get a nutrient molecule like glucose, which has energy associated in the chemical bonds, and then our cells will break those bonds, harvest the electrons to get the energy. In cells, energy tends to be obtained by catabolic reactions, and the energy is stored in the chemical, organic chemical compound that we're engaging in. And then that energy is taken and then used to make ATP. So cells make ATP for the purpose of using the ATP whenever the cell needs energy. It tends to get that energy from ATP. Because energy needs, well, because ATP supplies almost all of the energy needs in a cell, we say it is an energy carrier of cells, and we call it the energy currency of cells. Because when the cell needs energy, it doesn't use money to get the energy. Instead, its energy currency is ATP. It obtains its energy from ATP. Any question about any of that? All right, I once mentioned this before. When the cell needs to obtain energy from ATP, it breaks down the ATP. There is a high energy, unstable chemical bond in ATP, and it is the third phosphate group, which is generally broken to obtain the energy from ATP. The third phosphate binding is a high energy bond. And the second phosphate group is also bound to, well, that would be DTP, not adenosine DP, so adenosine ADP, adenosine diphosphate. Anyways, that bond also is a high energy bond. And if the cell needs more energy then it can get from just one ATP. It will then break ADP, and then we break off 2 phosphate groups from ATP. So before we talk about aerobic respiration, we need to talk a little bit about oxidation reduction reactions. And that is cells, when they're trying to produce energy, they frequently go through oxidation reduction reactions to obtain the energy. Like for example, when we are burning burning glucose in aerobic respiration, we are engaging in oxidation reduction reactions to obtain the energy needs from glucose. In oxidation reduction reactions, oxidation is when the electron is lost. So A loses an electron, so A becomes oxidized. The other molecule gains the electron. Gaining electron is called reduction. So B gains an electron right there, gains the electron that came from A. So there we say B is reduced. Oxidation reduction reactions are almost always coupled, meaning you have one molecule which will be oxidized and lose an electron, and then the other molecule will gain the electron, and we say it is reduced, or that is reduction. How do you remember oxidation reduction reactions? Well, you can say Leo, the lion, goes grr. Leo, lose for L, E for electron, O for oxidation. So Leo, lose electrons, oxidation. Leo, the lion, goes grr, grr, gain electrons, reduction. Grr, gain electrons, reduction. Leo, the lion, goes grr. Here's an actual example. We have copper +1 plus iron, which is a +3, can go through oxidation reduction, where copper loses an electron. Electrons have a negative charge, so it moves from +1 to +2. Iron gains the electron, and this is iron 3+ charges. When we gain an electron, the iron will be reduced to 2+ charges. Any question about any of that? This is not a partial charge, by the way. This is a complete charge. All right, if there's no questions, let's move on. Oops. Oxidation in biological systems often involves the removal of a hydrogen ion along with an electron. Remember that a hydrogen atom is composed of one H+ ion and one electron, which has a -1 charge. So when we have oxidation in a biological system, one molecule will lose the electrons, but it tends to lose the hydrogen at the same time. And then the other molecule will gain the electron, and at the same time, it will also gain the H+ ion. So we say that oxidation reduction reactions in biological systems is often dehydrogenation. meaning the hydrogen follows the electron of oxidation reduction. Any question about any of that? All right, so let's say that biological redox reactions, or meaning biological oxidation reduction reactions, are used in catabolism to extract the energy from a nutrient molecule. And I should warn you that redox means oxidation reduction. This is just a shorthand name for oxidation reduction, redox. And there's the oxidation part. There's the reduction part with the R on it. So when we use glucose in aerobic respiration, we are oxidizing glucose, and then another molecule will be reduced, such as NAD+. It will gain an electron and a hydrogen ion. Highly reduced compounds are compounds that have many hydrogen atoms. These highly reduced compounds tend to be degraded in order to get oxidation reduction reactions. For example, when we're burning glucose, the glucose is a highly reduced compound, and it will be catabolized into CO2 and water. And how that's happening is by oxidation reduction reactions. So here we have glucose. You can see it has many hydrogens, C6H12O6, and it will be oxidized down into CO2 and water. Any question about that? All right, the purpose for catabolizing glucose is to use some of the energy coming off of glucose to make ATP. Another way of stating that molecules like glucose, which are highly reduced, you can say they have a large amount of potential energy in them. And if you don't know, glucose has a lot of energy in it. That's why if you were to light the glucose, if I were to light it in a beaker right here, I'd have to step back several feet because so much of the energy is being released, and a little bit of that energy is converted into heat. Any question about any of that? All right, if there's no questions, let's begin talking about aerobic respiration. Aerobic respiration is the process that many cells use to make ATP. And most of your cells can only make ATP from aerobic respiration. The only cells which do not make ATP from aerobic respiration are your muscle cells. Your muscle cells, when oxygen is becoming limited, because the muscles are working so hard and so rapidly, they use up all the oxygen. And then the muscle cell can get its energy needs from a process other than aerobic respiration. And I'm just going to mention it, we'll talk about it later. And that process is fermentation. In aerobic respiration, we have the overall chemical equation stated up here, and that is glucose C6H12O6 plus 6 oxygen molecules, meaning 6O2, and 38 ADP molecules and 38 inorganic phosphate molecules get together to convert the glucose into CO2 and water. And so the products are 6 CO2 molecules, 6 water molecules, and then we generate 38 ATP molecules. Any question about any of this? We're going to talk about aerobic respiration happening in your cells, but please understand that in all cells, aerobic respiration is pretty much the same. And we will talk a little bit about the bacteria that are capable of performing aerobic respiration, like E. coli is one. It engages in aerobic respiration to make ATP. One other thing I need to mention in this slide, aerobic respiration does have another name. It also is called cellular respiration, presumably because most of your cells can engage in cellular respiration to make ATP. And so this name sticks and it's used in the textbook. And so you do need to know both aerobic respiration and cellular respiration. Just know it means the same thing. I don't tend to use cellular respiration because the cell can engage in respiration, but it doesn't have to be aerobic respiration. This isn't your cells, but certain bacteria can perform anaerobic respiration, in which case the glucose is not converted into, let me think about that. Yeah, I was about to tell you something wrong, so let me just stop and say that we will talk a little bit about anaerobic respiration later. The only way anaerobic respiration differs from aerobic respiration is mainly where the electron ends up. In aerobic respiration, the electron will eventually flow to oxygen. And so we say oxygen is the final electron acceptor in aerobic respiration. The only other difference that I know of is that the number of proteins engaging in that are responsible for aerobic respiration, if the cell is engaging in anaerobic respiration, it has the same, what do I want to say? It'll have the same enzymes and the same processes, but the number of proteins engaged in anaerobic respiration is fewer than the number of proteins engaged in aerobic respiration. All right, any question about any of that? If not, let's talk about how cells generate ATP. So some of the energy from a redox reaction, meaning an oxidation reduction reaction, is used by the cell to make ATP. Let me warn you that not all redox reactions can be used to make ATP. In fact, in our cells, there are only 2 chemical reaction, or I should say actually processes, that can make ATP. And that is fermentation, that we'll talk about later in this lesson, and then aerobic respiration. Both of these processes can obtain some energy from the molecule it's degrading, and we're just going to talk about glucose, to make the ATP. Some cells can make ATP by generating the ATP by phosphorylation. And then what we say is that ADP becomes phosphorylated, where we add a phosphate group. And this takes in energy because there's more energy in ATP than there is in ADP. So we have to supply energy for this reaction to occur. But then ATP can be generated. Any question about any of that? Let me point out to you, I already mentioned that in ATP, this third phosphate group is attached to ADP with the squiggly line. The squiggly line means this is a high energy covalent bond. And when we break this bond, we release a lot of energy. The same with this ADP, this second phosphate group is held to the first phosphate group with a high energy bond. And if the cell breaks the first phosphate group off of ATP, but still needs more energy, it can actually break the second phosphate group off to obtain more energy. Any question about any of that? I think I already mentioned, but maybe not today, and that is we add inorganic phosphate to ADP and energy to make ATP. All right, this slide is showing you the overall summary equation. Let me shrink that down. You should know the summary reaction for aerobic respiration. Oh, I went the wrong way. There are three mechanisms cells have for making ATP. The first mechanism is substrate phosphorylation, where we transfer a phosphate group to a molecule like this molecule here. And we transfer this phosphate group from this molecule to ADP, generating ATP. This is substrate level phosphorylation. This is how we generate ATP in fermentation. ATP is made in fermentation by substrate level phosphorylation, where we directly move the phosphate from one molecule to ADP. This reaction does take energy, which isn't shown here, for this reaction to proceed. The second way cells have for generating ATP is by respiration. This can also be called oxidative phosphorylation, and your textbook occasionally uses this term. I'm only going to use respiration. Any question about any of that? All right, respiration, and you might notice I haven't said aerobic respiration, because when we're talking about respiration, we mean both aerobic respiration and anaerobic respiration. So cells can generate ATP by either anaerobic respiration or by aerobic respiration. How the cells generate ATP from aerobic respiration is that they sequentially transfer the electron, which initially came from glucose, from one electron donor to another electron molecule that we call the electron acceptor. And then that electron, which actually starts from glucose and then moves to NADH. NADH, you don't need to know this, moves to FMN, which then the electron moves to Q. Is that what that is? I can't read it. Which then moves to cytochrome A, or is that B, one of the two. Then moves to cytochrome C, moves to another cytochrome, moves to another cytochrome. And then that electron eventually moves from the last molecule to oxygen. So that we say the final electron acceptor is oxygen. How that electron moves is by oxidation reduction reactions. Let me blow this up a little bit. The two are always paired. When the electron is flowing from NADH to FMN, NADH loses the electron as well as the hydrogen. And so we say it is oxidized, LEO. And then FMN gains the electron, so it is reduced. And then FMN sheds the electron to Q, so it will be oxidized, and then Q will be reduced. And the point is, for all of these, the electron moves from one molecule to another, and along the way, we'll have numerous oxidation reduction reactions. The purpose for this is to have the electrons flowing from one molecule to another so that the energy being released from glucose isn't released all at once. It's released in a stepwise process. Because the cell, if you were to release all of the energy from glucose all at once, you would overheat the cell and the cell would die. As I mentioned, to get the energy released from glucose, you can take a blowtorch to it, and that tells you the amount of energy that's being released. Another example is, I don't know if I mentioned this one or not, I meant to, and that is how much energy is in glucose. Well, people who make homemade bombs, one of the ingredients that they can use to make a homemade bomb is actually glucose. Any reaction, question about that? The point is, glucose has a lot of energy in it, and we can use that energy to make a homemade bomb, for example, if you were so inclined. But more likely, you're going to be using glucose and obtaining its energy needs to give your energy needs. And we'll do that with mainly by aerobic respiration to get the energy from glucose to make ATP. The third way that cells can engage in making ATP is by photophosphorylation. This occurs only in photosynthetic organisms. What happens is light releases an electron from chlorophyll. That electron then flows down a chain of proteins. We'll talk about that. And then that electron movement is used to make ATP via a chemical reaction. And actually, it's going to use an electron transport chain. And we'll talk about that at the end of this lesson. How photosynthetic organisms use light to get their energy needs. In photophosphorylation, ATP and NADPH are usually generated. However, the cell often will use up the ATP as well as NADPH to make sugar. Any question about any of that? Oops, didn't mean to shrink it that size. Didn't mean to do it that way either. I'm trying to get it to normal size. I'm just having a hard time here. All right, here we see an example of photosynthetic organisms. That's why they're all muddy. They're in the sunlight and they're engaging in photosynthesis. Any question? All right, only kidding. They're not engaging in photosynthesis. However, the green plants in the background are engaging in photosynthesis to make their energy needs and generate ATP by photophosphorylation. So there are more than one metabolic pathway to get the energy needs of a cell. At this point, we're only going to talk about one, and that is catabolism, the burning of glucose to make ATP. Although we did mention one just briefly in the last slide, and that is photosynthesis can generate ATP by using light, and that is not from a catabolic reaction. The energy that's released in this catabolic reaction is released in controlled ways and is stored by a series of chemical reactions, meaning the energy is not released in a single energy burst. Electrons from donor molecules are passed from one compound to another through a series of redox reactions. And there we're seeing most of the oxidation reduction reactions that are used to make ATP. You don't need to know that. You don't need to know any of this, although you should know the start maybe NADH is where the electrons come from when NADH is carrying the electrons from glucose to NADH. And then the other thing to know is the electrons will eventually end up at oxygen in aerobic respiration. And that's why we say the final electron carrier is the final electron acceptor is oxygen in aerobic respiration. Many organisms can use carbohydrate catabolism for obtaining their cellular energy needs. And these organisms engage in catabolism, but they tend to burn carbohydrates. The most common carbohydrate that is used in carbohydrate metabolism is glucose. So the most common energy source is glucose, but it's not the only energy source. For example, if you were to eat a diet rich in pasta or carbohydrates, it's not going to contain much glucose, but will contain starch, and you will eventually go through a carbohydrate catabolism to burn the starch, and the starch will be converted into glucose, and then the glucose will be burned in aerobic respiration in the normal way. And that's why cells tend to use carbohydrate catabolism to get their energy needs. And that is the carbohydrate will be converted easily into glucose, generally. And then glucose will be burned in aerobic respiration to generate the ATP. Okay, that's not exactly correct with like fructose. it's a little bit more complicated. And then the fructose has to be converted into glucose. But once again, that will be the ultimate step, convert the fructose into glucose, and then engage in glucose burning and aerobic respiration to obtain our energy needs. Carbohydrates are not the only way cells can do this. They may also use lipids and proteins. These, however, are not the preferred molecule for engaging in aerobic respiration. For example, a marathon runner can burn lipids in aerobic respiration to get their energy needs. But a marathon runner will not burn lipids until after 45 minutes of the run. So obviously this is not a sprinter, because a sprint is not usually longer than 45 minutes. This would be a marathon runner, can actually start metabolizing lipids to get their energy needs. But that doesn't happen until 45 minutes until the run. And what the runner, the marathon runner, is first burning is they're first burning carbohydrates to get their energy needs. And yes, people can burn proteins, get their energy needs. This happens to people who have a high protein diet. They don't have enough carbohydrates to burn, so they'll start burning proteins. Any question about any of that? All right, so to produce energy from glucose, microorganisms use two general processes. They use respiration and they use fermentation. And I should say that your cells can also do this using respiration and fermentation. All of your cells can get their energy needs from aerobic respiration, but your muscle cells can get their energy needs from aerobic respiration. Any question about any of that? All right, that's your cells. We're not just talking about your cells. We're also talking about microbes. Microbes can get their energy needs from respiration and fermentation. If they get their energy needs from respiration, they can get it from aerobic respiration, or they can get it from anaerobic respiration, depending on what the cell is. Your cells, none of them can get their energy needs from anaerobic respiration. But bacteria, for example, like E. coli, can get their energy needs from anaerobic respiration. E. coli can get their needs from aerobic respiration. Sorry, I didn't mean to do that. Whenever oxygen is available, E. coli will use aerobic respiration. But if oxygen is not available, E. coli can get their energy needs from anaerobic respiration. E. coli can also get their energy needs from fermentation, and we'll talk a little bit about each of these processes that the cell can use to make ATP. We'll talk about aerobic respiration in great detail, and we'll talk about that first. Let's watch a little video on aerobic respiration. Where the heck is it? Oh, there we go.
All cells need energy to stay alive. Usually,
Oop, wrong way. Let me turn that up. Let's turn it up here too. No, that's pretty good actually there. Go back a little.
alive. All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions? So let's talk about ATP. Not ATP. Let's talk about aerobic respiration. This slide is showing you a little bit of a summary about what we're going to be doing. We're going to be talking about aerobic respiration next, and then after that, we'll talk about fermentation. Both tend to burn glucose, which goes through glycolysis. In fermentation, we then have a fermentation reaction, or 2 that occur after the glycolysis. In aerobic respiration, we have glycolysis, which is the first step of aerobic respiration. Then we have the preparatory step in the Krebs cycle, the next step of aerobic respiration. And we have the electron transport chain, or ETC, which is the third step in aerobic respiration. I should mention that I'm going to discuss this for aerobic respiration, but actually anaerobic respiration tends to have each of these same 3 steps: glycolysis, the Krebs cycle, and the electron transport chain. Essentially, in aerobic respiration as well as anaerobic respiration, the electrons are going to be flowing from glucose, which is an energy-rich molecule. And these electrons are going to flow to oxygen. The glucose will be converted into CO2. Water will also be made in this process. And the CO2 and the water are energy-poor molecules. The glucose is an energy-rich molecules. We're getting the energy from glucose, and we're using that energy to make ATP. Aerobic respiration has a high ATP yield. Any question about any of that? All right. In fermentation, we'll talk about this later. The electrons flow from glucose and they actually flow to a molecule of the molecule of fermentation. So they're going to flow to pyruvate, and the pyruvate will be converted to the end product, which will often be ethanol or lactate. And ATP is generated. However, there's a much lower ATP yield in fermentation compared to aerobic respiration. Any question about any of that? All right, if there's no questions, let's move on and start talking about aerobic respiration. Here is a overview of aerobic respiration in a eukaryotic cell. The purple will be the cell, and then the mitochondria is shown here. Aerobic respiration begins with glycolysis. This occurs in the cytoplasm of the cell. Some ATP is made by glycolysis. We'll talk about NADH later. But some is made in glycolysis. The glucose is converted in glycolysis after a number of steps into pyruvate. Pyruvate then moves into the mitochondria and will be used by the Krebs cycle. Here it's called the citric acid cycle, and there's multiple names for that. I'll call it the Krebs cycle. And the glucose will eventually be converted into CO2. Okay. The Krebs cycle happens inside the mitochondria of a eukaryotic cell. The Krebs cycle will then generate, will generate some ATP shown here, but it will generate a number of NADH molecules and some FADH2 molecules. The NADH and the FADH2 molecules from the Krebs cycle and from glycolysis will take the electrons, which are being carried by NADH and FADH2, to the electron transport chain, which is shown here, which has also got other names like oxidative phosphorylation. I won't call it that. I'll always call it electron transport chain, which is what your book calls it too. It's also called chemioosmosis. Like I said, there's a lot of names for this. I'll always call it the electron transport chain. And here the electrons are passed to the electron transport chain where ATP is produced in large amounts. Now, this is showing you what's happening in a eukaryotic cell. It's a little different in a prokaryotic cell, and that is we still have glycolysis happening in the cytoplasm of the cell, if the cell is a prokaryote. However, we don't have a mitochondria in a prokaryotic cell. Where the electron transport chain is happening is in the cell membrane, in and along the cell membrane. Any question about any of that? All right, let's go on and talk about aerobic respiration. The first step being glycolysis. Glycolysis has another name, which I will never discuss, Embden-Meyerhof pathway. named after the people who discovered it. In glycolysis, you have the oxidation of glucose to form pyruvic acid. Glycolysis does occur in most living cells. It can occur in the presence of oxygen, but it can occur in the absence of oxygen. It does produce some ATP and some NADH. What did I want to say about that? Oh, yes. Shown right here. Well, I'll go to the next one. We start with glucose in glycolysis, and we end up with pyruvic acid. Trying to blow this up. Glucose is a 6-carbon molecule. We generate 2 pyruvic acid, shown right there, in glycolysis. And that's because pyruvic acid, or pyruvate, is a 3-carbon molecule. Any question about that? So because there's 3, there's 2 pyruvic acids that are generated by glycolysis. Let me also state that we will use pyruvic acid and pyruvate as synonymous. If you were a chemist, you wouldn't like that. Pyruvic acid has the hydrogen atom attached to it. Pyruvate has the hydrogen atom released. But other than that, we are the same. And the cell uses those molecules the same. So we're going to use those 2 terms interchangeably. In glycolysis, it's about a 10-step process shown here, where the glucose is converted into, see if I can blow that up a little, into glyceride 3-phosphate, or dihydroacetone phosphate. And then the dihydrogen phosphate is converted into glyceride 3-phosphate. So it goes down this process twice. When I was an undergraduate, I had to know the name of each intermediate as well as each enzyme that made the intermediate for all of glycolysis. So because I was required to know all of the intermediates, I think it's fair that you guys are going to be required to know all of the intermediates, meaning memorize this slide. What do you guys think? You guys think it's fair? What did I just do? Okay, so you guys have to memorize this slide. Know that there are 2 ATP that come into glycolysis at the start, but we generate out 4 ATP. So we have a net gain of 2 ATP from glycolysis, starting from glucose.
Would you like us just to be able to label the steps, or be able to like write it out in paragraph form as well as what's happening?
You guys weren't commenting, so that's why I was saying you're going to have to memorize each of the steps, because I had to know each of these different steps and all of the enzymes, which isn't even shown here when I was an undergraduate. But no, you guys don't need to know that. You just need to know the summary reaction shown here. You should know the glucose is starting the process, you generate 2 pyruvates or pyruvic acids. You get a net gain of 2 ATP. Just remember, you put in 2, get out 4, so that's a net gain of 2. And then you generate 2 NADH. This is all you need to know. What is in red, and that glucose starts the process. Okay, any question about any of that? All right. After glycolysis, we then have the electrons flow to the Krebs cycle. And what happens is the pyruvic acid moves to start the Krebs cycle. Let me take a step back and state that respiration doesn't have to follow aerobic respiration, where the final electron acceptor is oxygen. That respiration can be anaerobic respiration, which microbes can, a number of them can perform anaerobic respiration. And the final electron acceptor is a molecule other than oxygen. It might be nitrate or a sulfate or an iron molecule. It can be many different molecules, but it's something other than oxygen. ATP is generated. All right. Pyruvic acid is what was made in glycolysis. And the next step is the Krebs cycle. However, pyruvic acid cannot join the Krebs cycle. It has to go through the preparatory step first, which is a 1 or 2 step process where pyruvic acid is converted into acetyl coenzyme A. Pyruvic acid is a 3-carbon molecule. Acetyl coenzyme A is a 2-carbon molecule. So 1 carbon dioxide comes off of pyruvic acid to make acetyl coenzyme A. We do generate 1 NADH in the preparatory step. And now the acetyl coenzyme A can join the Krebs cycle. Any question about that? All right, no questions. Let me state that the preparatory step runs 2 times for each molecule of glucose. Why does the preparatory step run 2 times for each molecule of glucose?
Because the glucose molecule is cleaved in half.
Yeah, it is cleaved in half, and you get 2 pyruvic acids made from glycolysis. So the preparatory step will run twice for each molecule of glucose. All right, the next step is the Krebs cycle. The Krebs cycle is also called the tricarboxylic acid cycle, or TCA for short, for tricarboxylic acid. And it's also called the citric acid cycle, which is also abbreviated TCA for the citric acid cycle. So it's all of these names. I will always call it the Krebs cycle. Your book at least tends to call it the Krebs cycle also. And that's after Dr. Krebs, who discovered it. Any question about any of that? So here we're seeing the preparatory step, converting the pyruvic acid into acetyl coenzyme A. The acetyl coenzyme A then binds to, let me get this out of the way. Try and blow this up. Still on the way. Well, we're gonna have to have it in the way. The acetyl coenzyme A binds to oxaloacetic acid, which is then converted into citric acid. Citric acid is made into this compound. That compound is made into this compound. This compound is made into that compound. This compound is made into that compound. This compound is made into that compound. That compound is made into that compound. That compound is made into oxaloacetic acid, which is where we started. Why it is called a cycle. You end up in the same place where you began. Any question about any of that? So the Krebs cycle will always run as long as we have acetyl coenzyme A coming in. For the Krebs cycle, we generate one ATP, which is made here at this step. We generate 4 NADH, one in actually the preparatory step right here, one right here, the second, the third right here, and then the fourth right here. So 4 NADH are made in the preparatory step and the Krebs cycle. We also generate 1 FADH2 right here. And then we generate 3 carbon dioxides. Right here in the preparatory step is 1. The second is right here. The third is right there. So all of the carbons in pyruvic acid are converted into CO2. 3 CO2 is coming out of the preparatory step and the Krebs cycle. The NADH and the FADH2 go to the electron transport chain. They're moving the electrons, which came eventually, or what do you call it, originally from glucose. They will be carried by NADH and FADH2 to the electron transport chain. Okay, so you need to know that what is shown in red is what happens in the preparatory step and the Krebs cycle. You should also know that pyruvic acid begins the preparatory step and then it ends with, I guess, CO2. And you generate 4 NADH and 1 FADH2 and 1 ATP along the way. Any questions about any of that? All right, how many times does the Krebs cycle run for each molecule of glucose?
2 times.
2 times is right, because there will be 2 pyruvic acids made in glycolysis from 1 molecule of glucose. So be careful when I ask you a quiz question. If I ask you how many ATP are generated by the Krebs cycle per molecule of pyruvic acid, or if I ask you how many ATP are generated in the Krebs cycle per molecule of glucose. Because the answer, if it's pyruvic acid, is 1, shown here. But if it's glucose, it'll be 2. Any question about that? Okay. And to get how many you have in glucose, you just multiply all of these numbers by 2. All right. You do not need to know oxaloacetic acid, but you should realize that we end up here, and this is where we start, why it is called a cycle. that this goes in a cycle. Come on. All right. So after the glycolysis and the preparatory step in the Krebs cycle, we then enter the electron transport chain. And how that's happening is the electrons from glycolysis and the preparatory step in the Krebs cycle are taken to the electron transport chain by NADH and FADH2, which FADH2 is not shown here. Any question about any of that? How are we doing time-wise?
Our lab was supposed to begin at 7.
Oh, you guys, at 6:50, you need to tell me to stop, okay? All right, let's go ahead and end this. And we'll begin the lab at 7:10, okay?
Thank you.
You're welcome.
Thank you.